So going off of what Taylor mentioned in Figure 3D, the results indicated that scavenger receptors were the receptor utilized by Dons in a kind of receptor-mediated endocytosis. Now, this partially answers the question of how the Dons were taken in by the cells, but not exactly the mechanism of how it occurs. And this information is not fully verifying that scavenger receptors are at play here. So, also referencing what Nicole said earlier in Figure 1, AUNP barcodes, gold nanoparticles, were arranged and bonded about 5 nanometers apart on the large rods. The reason for the large rods, there were several. One is that it was found to be the most efficient Don in cellular uptake, making it a prime variable for the testing. But also, the AUNP, the gold nanoparticles, are readily taken in by cells. So they wanted to be sure that it would be a Don large enough and linear enough to have a uniform pattern. That's where you see the barcode linear pattern here. Easy to identify through transmission electron microscopy, or TEM imaging. You can see it was actually a test run counting the different numbers of gold nanoparticles on each large rod Don, and it was found that on average 6 or 7 little particles were on each Don through transmission electron microscopy and through gel electrophoresis. This was all to run this test of cellular uptake with these gold nanoparticles so that they could be viewed by transmission electron microscopy. So when you see just these 6 black dots, it is just showing the gold nanoparticles, but it can be assumed that these are in fact the large rod Dons. And it is going to be going through, supposedly, this proposed mechanism here. This mechanism is actually a mix of 2 other mechanisms that that the test is kind of working to identify and discern between. So one is clathrin and the other is caveolin dependent. This is a material that ends up binding to the membrane folding and kind of determines where the materials inside end up in the cell. With clathrin dependent, it is going almost definitely directly to the lysosome. The significance of that is that with the lysosome, it gets broken down to its base nutrients to be used throughout the cell. Whereas with caveolin dependent mechanism, it will end up from an early endosome to a late endosome, wherein it could go to a more targeted area of the cell. This could be the endoplasmic reticulum, the Golgi apparatus, even the cell nucleus. So it's really a question of how selective the Dons are in terms of their pathway through the cell, in terms of drug delivery. So moving on, you can see in the first stage with the TEM imaging, the transmission electron microscopy imaging, you can see the large rod interacting horizontally with the cell membrane. This is where it is assumed that the Dons are interacting with receptors, the scavenger receptors specifically mentioned by Taylor. You can see that it's interacting horizontally, possibly just explaining why the large rod is more efficient than a more three-dimensional Don or a smaller Don, given that it has more surface area and a greater ease of endocytosis taking it in. There's, less space that it takes up in general. For invagination to occur, that is what is occurring here. You see an endocytic kind of folding of the cell membrane, and this is what makes up the process of endocytosis, taking in the taking in the DONs. Here you can see that the structure was pretty much fully maintained. You can see 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7 in agreement with the previous figure mentioned on gold nanoparticles. Here you see basically several blobs. These blobs are supposed to be early endosomes because you can tell that there are some full structures, possibly even this one, remaining, indicating that the DONs were not degraded in this process. But you can see some gold nanoparticles scattered about, which would lead to the assumption that there is some breaking down of the DONs in terms of their structure. Here we could see a full degradation of the DON structure, assumably because you just have a blob of gold nanoparticles. This is what would be assumed to be within the late endosome or the lysosome, depending on whether it is a claviolan or clathrin dependent, respectively. The only issue is this cannot be determined through this particular test with TEM, largely because the late endosome and lysosome both have highly acidic environments that are acidic enough and have enzymes to break down the DONs to equal efficiency at that point in time. So it's not definite at this point which pathway the DONs took. And this means that, as summarizing here again, in each process you can see throughout these images verifying that it's the same pathway more or less each time. Endocytosis after interacting horizontally with the cell membrane, indicating a receptor-mediated endocytosis. But the type of receptor-mediated endocytosis is not known yet. It is most likely scavenger receptors as they interact with nucleic acids, and that was verified earlier in 3D, but it's not known which pathway inside the cell it takes. And this is important because the selectivity of the DONs kind of dictates which drugs can utilize DONs as a vehicle to enter cells, depending on where in the cell that drug needs to end up and how intact that drug needs to be in order to do its work on the cell. So in figures 1, 2, 3, and 4, the results of several tests were shown. All of these focus on how viable DONs are as a vehicle for targeted drug delivery to cancer cells. First of all, there was an initial comparison of DONs in terms of size and structure. Modifications were made to determine if this would affect cellular uptake. First of all, they had to determine if these would survive in a cell-like environment. They did. They then worked to determine if they would be efficiently taken in by two different lines of cancer stem cells, the H1299 and the DMS56. In both cases, all of the DONs were successfully taken in by the cells, although H1299 had a higher rate of cellular uptake, and the larger cells in this and the larger DONs in both this set of trials and in another set of trials with just the H1299 over a span of time showed that the larger DONs, the larger DNA origami nanostructures, were significantly more efficient than the smaller DNA origami nanostructures, possibly as indicated by the TEM imaging, due to more surface area to interact with the cell membrane. The question at this point was asked of how exactly they're getting into the cell, and it was determined by another inhibition study of scavenger receptors that scavenger receptors, were responsible for the uptake of DONs. Beyond this, they wanted to know how exactly this is occurring, and determined that yes, it is validated further that yes, this is receptor-mediated endocytosis, but still remained with a question of whether it is clathrin or claviolan independent, how targeted the transport of this DON is. And it was determined, of course, that they do get fully degraded once they enter the cell. With another time trial run of timed exposure over like hours and hours of time of cancer cells to DONs, it was determined that the DONs were able to be steadily taken in by the cells for times as long as 72 hours, and were still found in traces after 96 hours. That's over 3 days. All of this indicates that DONs are able to survive in a cell-like environment and in a living cell environment, are able to be successfully taken in by cancer cells, and do this by receptor-mediated endocytosis. It was found that they could remain in the cell for a long period of time and could also be administered over a period of 3 days with steady cellular uptake. All of this validates DONs as a vehicle for assisted drug, for targeted drug delivery to cancer cells, kind of supporting the hypothesis generally speaking. There's still, of course, further questions which should lead to further research, however, namely whether DONs are inherently better than other nanostructures or other known chemotherapeutic drugs that are being used, whether they are how necessarily they measure up. Now, this could be a similar experiment to the time trial run with several different DONs. It could take the large raw DON, which was considered the best, and compare it against several other known vehicles for targeted drug delivery in cancer cells. Speaking of targeted drug delivery, a test should probably be run comparing the effectiveness of the DON with healthy cells, along with cancer cells, and even cancer stem cells as well, if possible. All of this to verify whether the DONs are actually targeted, and also whether whether this will impact anything else. I'm sorry, I lost my train of thought. There could also be a further test run comparing the overall cytotoxicity of other drugs utilizing DONs, or compared to the drug on its own, to see if the DONs are really impeding or helping the process of specific drugs. But that would have to be on a case-to-case basis in determining if the DON is a viable vehicle for any specific drug. Thank you.